The last portion of this neuroanatomy lecture is going to be devoted to sensory anatomy. So the anatomy of the sensory system. We spoke about the central nervous system pathways, but now we need to talk about more peripherally what we have as far as sensory anatomy. So the senses can be divided into general and special. The general senses are senses such as temperature, pain, touch, pressure, and vibration, as well as proprioception. The special senses are senses such as smell, taste, balance, hearing, and vision. So the receptors for these different modalities are cells or specialized cells that are named after the sensation they respond to, they detect. So if you look at all these names, they're actually very intuitive. Just by the name, you can get to the sensation that they are detecting, except for one or two. So let's start with exteroceptors. Exteroceptors is a collection of receptors that are detecting information about the external environment. For example, you grab a mug to have your coffee, and you detect, for example, temperature. You could also detect if it's dry or wet. You could also detect even if you step on something painful. So all those are sensations that come from the outside. Interoceptors, on the other hand, are capturing something about inside of the body. Proprioceptors carry information about proprioception. It's difficult to know what proprioception is just by defining it. The definition would be something like the position of the body. But what helps a lot is to think about a clinical task that allow us to test for proprioception. So you can ask your patient to close their eyes, and then you lift the finger, for example, of the patient and ask him or her whether the finger is up or down. So see, the patient without using the eyes can tell whether the finger is up or down. This is what proprioception is allowing a person to do in this task, and therefore helps us understand. Thermoreceptors are for temperature. Mechanoreceptors are for a physical distortion, so anything that moves can be considered mechanoreceptors or can be detected by mechanoreceptors. So we can speak about mechanoreceptors as being receptors that are used for touch, for pressure, for vibration, for example. And then we have chemoreceptors, which are for chemical senses. So senses that depend on chemical substances are senses such as the smell and the taste, for example. Finally, I left this one to the last because it is a little bit not intuitive. Is nociceptors. Noci stands for pain, so these are receptors for pain. So we are going to start this section by speaking about the general senses. So the general senses are going to be detected then by a series of receptors. The simplest form of receptors are what you call free nerve endings. representing that the neuron itself has free nerve endings that work as a receptor. The free nerve endings are oftentimes receptors for pain, so they are nociceptors, but not only for pain, but also for temperature, for example. I will ask you here to think about two different pains you have experienced. For example, think about the pain that you experience when you step on a sharp object. Now think about the pain you experience the following day if that area of the body is sore. So these two pain sensations are different from one another. One is fast and acute, one is slow and feels more like a soreness. So it can be tempting for us to understand these two modalities based on the brain only. So we could understand and we could easily describe certain sensations as evoking a more complex mental stage. So we can think that we could be afraid of needles, and needles therefore capture our attention. And those aspects may play a role, so those psychological aspects and more central nervous system aspects. However, peripherally, it's important to understand that we have two kinds of axons that are responsible for carrying pain signals. We have axons that are myelinated and axons that are non-myelinated or unmyelinated. And we spoke about myelin before, and myelin allows the axon to propagate the signals faster and more efficient. So you have here two types of fibers, axons. So we have the type A, and you have the type C. The pain type A we can even add a little bit more, and it's called type A delta. So whether the pain stimulus is stimulating a type A, a receptor that is connected to a type A fiber, or a type C fiber, the pain is going to be processed very differently. So we have fast pain, a pain that the signals are carried by type A fibers much faster, and the slow pain in which the signals are carried much slower. So it's a more of a, sometimes people call this more of a chronic pain, although clinically chronic and acute have more the sense of the duration of the pain. And that's pretty much what I would like you to understand about pain. So talk about nociceptors. In particularly, I would like you to think about the free nerve endings as one kind of nociceptor. Then the signal is carried to the central nervous system through either fast pain fibers, which is the type A fibers, or type C fibers, which give rise to the slow pain. Then the other pathways you already know. You already know that this pain, temperature, and light touch are going to be carried into the spinal cord in a certain way. So you have to connect all that, and the pain sensation will go into then the signals. The second order neuron will cross, it travels to the thalamus, and from the thalamus a third order neuron projects to the somatosensory cortex. So you can put all together, and then when you see those diagrams that we introduced before, they'll make more sense, because now you know all the pieces to understand those diagrams. There is, however, something else that I want to talk about pain, that is the case of referred pain. Referred pain has to do with the pattern of innervation. So viscera, the inside organs don't have a very defined innervation, and when there's a painful situation affecting those organs, they will borrow the somatic nerves. So we will feel pain in certain areas of the body. They are more somatic, more external, if you will, but in fact the pain comes from a different organ. That's the concept of referred. The classical example is the heart attack. A heart infarction borrows, the pain will borrow C fibers, so the spinal roots of C nerves, and therefore the pain can be felt all the way from the arm to the neck. It won't be felt above the neck because that's territory of the trigeminal, so it doesn't go as far. Other components, for example, the liver and the gallbladder, that give rise to a referred pain on the right shoulder, and even the pain of the stomach, small intestine, and colon are referred because they don't match the anatomy of the organ. So we move from pain. We finished pain, the anatomy of the pain receptors here, and now we move to mechanoreceptors. Here you'll need to know all the different kinds of mechanoreceptors that exist. But before that, I want you to, before we go into the names and so on and so forth, I want you to emphasize again or repeat again that mechanoreceptors respond to some sort of movement of the body. So we talk about stretch, we talk about compression, twisting, or distortion of the cell membrane. What is important to understand, but again, I would like you to just pay close attention to the names and know a little bit about it, but you're not supposed to know a lot, is that there you have the areas of the skin. So you see the middle picture, but I will show you more. I will show you a better picture in just that zooms in on all this. But what is important to understand is that the receptors are in different depths. So some are more superficial, some are more are deeper. So this will be capturing whether the distortion is very superficial, like what happens to when we are carrying, for example, a light object, versus when the distortion is very deep. For example, the distortion caused by a heavy object lying on our hands, for example. So that's the location is very important. Then physiologically they are they vary from one another in how fast they adapt. So some receptors keep responding to the stimulus, others stop responding once after a while. But I'll leave that for you to learn in physiology with more detail. But so these mechanoreceptors work in very different sensations. We'll focus on the tactile, so on the touch, but we can also use mechanoreceptors for baroreceptors, so receptors for blood pressure, for example, although there's other pressure receptors, for example, the distension that our bowels have when the feces are there. So all those are our baroreceptors, sometimes called stretch receptors, and proprioceptors also, again, that has the position of the muscles and the tendons and the bones. So the first component of the tactile receptors are the free nerve endings. The free nerve endings are what we call unencapsulated tactile receptors. We spoke about the free nerve endings for pain. The free nerve endings may also detect other sensations. So you see They are very superficial, and they are a continuation of the neuron. One special kind of receptor has to do with receptors that are wrapping around the hair follicle. So these receptors are detecting distortions that have to do with the hair. These are called root hair plexus. Then we have still unencapsulated tactile receptors. We have the free nerve endings synapsing with Merkel cells. So the Merkel cells are sensitive to light touch. So understand they are superficial, and understand that they are sensitive to light touch. So light, you can see touch being light or heavy by what object you're touching the person with. So you're touching a person with a piece of cotton, it will distort just very lightly the skin, and is captured, for example, by the Merkel cells. If you're touching someone with a heavy object, the object will cause a greater distortion that it needs to be sensed by deeper receptors. So these are the three unencapsulated tactile receptors. Moving now to receptors that have a capsule, we encounter the tactile corpuscles. Note that they are superficial, and they adapt fast, so they stop responding fast, and therefore they are sensitive to initial contact. And you can find them in the eyelids, lips, fingertips, nipples, and external genitalia. Going a little bit deeper in the skin, we find the bulbous corpuscle. The bulbous corpuscle is also named after the anatomist, and is called Ruffini corpuscle. This is deeper, and it's going to be associated with a distortion and stretching of the dermis. So we are going deeper. So again, this would be for a deeper distortion. And finally, the last encapsulated tactile receptor is called a lamellar corpuscle. A lamellar corpuscle is made of layers, several layers, and is also sensitive to deep pressures and to high frequency vibrations. So again, you are expected one or two questions about these receptors, because it's a kind of knowledge that you either learn it in anatomy or in a cell biology class, or you may never you may never really force yourself to learn these. So it's important to really pay attention to these receptors at least once, so that when you go to physiology, you just accept that you understand that the receptors vary in terms of depth, the receptors, and then you add more layers to it, such as vary in terms of how fast they adapt, and for example, what kind of vibration they respond to. But that's something you can add to physiology. For now, I'd like you to know very well the depth and what kind of sensation they are responding the best. Moving to baroreceptors, those are oftentimes called stretch receptors, and they'll be detecting pressure. Pressure where? We have spoken about the glossopharyngeal nerve and how the glossopharyngeal nerve captures information regarding the carotid sinus. That information, we will speak at some point about chemoreceptors, but some of that information is also baroreceptors. There are baroreceptors in many organs that can distend, they can stretch, such as the lungs, the digestive tract, and the urinary bladder. So we already spoke about this when we spoke about the muscles, but it's important to understand that the muscles and the tendons have receptors. These are the exact receptors that provide us with proprioception. So we have the muscle spindles inside of the muscle, and we have the Golgi tendon organs for the tendons. The muscle spindles detect changes in muscle length and changes in the rate of the change of the length. The Golgi tendon organs do a comparable task, detecting changes of tension and detecting the rate of change of tension for the tendons. Finally, the chemoreceptors detect chemical substances. We will speak about the taste and the smell, which are special senses, but there are chemoreceptors that detect, for example, changes in the pH, CO2, and oxygen, which are going to be important. That information is important for changes in the breathing rate. The peripheral chemoreceptors detect pH, CO2, and oxygen. The central chemoreceptors that exist in the brainstem detect only pH and CO2. The peripheral receptors are in the carotid body and in the aortic body. So now we can move to special senses. Here it's we're going to focus on the anatomy, but it's inevitable that we can see it all as one function. So we can answer the question, how do we smell, or how do we sense a taste, or how do we perceive vision, or how do you sense sounds? So we'll start with olfaction, the smell. We covered this already when we talk about the olfactory nerve, but here is just to really show you what the olfactory sensory receptors look like. So you see that they are in the top of our nasal cavity, so require that we breathe in through our nose to have a better sense of smell, and then the olfactory nerve fibers that pass through the cribriform plate and form the olfactory bulb, the olfactory tract, and then they project into the nervous system, the central nervous system. Gustation or smell, these are sensed by the tongue. The tongue has papilla. There are different kinds of papilla, so we don't have to worry about the shape of the papilla, but each papilla has what you call a taste bud. So you see what a taste bud looks like to the right. So representing that when the chemical substance stimulates this taste bud, the taste receptor cells are going to be activated. So we have 5 tastes. We have salty, sweet, sour, bitter, and umami. So some textbooks give us the impression that there's a map for the taste in the tongue. Certain areas are for one taste, other areas are for other tastes, but that's not actually accurate. It's a very widespread idea, but not actually accurate. The tongue, you can see the distribution on the right side, and we have receptors for all the 5 tastes throughout the tongue. So moving to equilibrium and hearing, we need to now know the anatomy of the ear. The anatomy of the ear is very simple, and this is the picture. You have to know all the structures that are labeled in this picture. But there is a functional aspect to it that we can add to make this story more interesting and simultaneously to know more about the physiology of hearing. So you see in this picture the division between an external ear, the middle ear, and the internal ear. The external ear consists of the auricle, sometimes called the pinna, the external acoustic meatus. So as I say the words, please follow them and pause. Pause if you need some time to find the label, because I cannot point. The external acoustic meatus, and then ends in the tympanic membrane. This external ear is basically allowing the amplification and the funneling of the sound, which involves movement of particles, so the sound wave into the tympanic membrane. The tympanic membrane, from the tympanic membrane in, we start the middle ear. The middle ear is filled up with air and in what's called the tympanic cavity. And we have 3 ossicles, tiny little ossicles, the smallest of the bones in the body, which are the malleus, the incus, and the stapes. I have a slide for you to zoom, in which I'll zoom in the auditory ossicles, but I still would like to cover more details. The middle ear is connected with the pharynx through the pharyngotympanic tube, or sometimes called auditory tube, or sometimes even the eustachian tube. I cropped this picture so you don't see very well the tube, but I'm going behind to show you the tube clearly here. In addition to the auditory tube, there are also 2 muscles, the tensor tympani and the stapedius, which are not clearly seen here, but I'll show you in a little bit where they are. So here are the muscles. So you see the tensor tympani and the stapedius. And in this slide, I'm also showing you the auditory ossicles, the malleus, the incus, and the stapes. Finally, we go into the internal ear. So in the internal ear, we have a bony labyrinth of the internal ear, and inside we have a membranous labyrinth. We'll have the receptors for the sound, which are actually the hair cells, and you have fluid circulating, which is the endolymph and the perilymph. So the vibration is passed onto the cochlea, and the goal of this vibration is to change activity of the hair cells. So let's see how this happens. So this is what a cochlea looks like. So you see that outer part is bone, and the inner part is the membranous labyrinth. The cochlea has two openings. One is called the round window, the other is called the oval window. The round window is for the exit of the axons that form the nerve. The oval window is for the connection between the stapes and the cochlea. So what does it look like inside? So here you have the cochlear structure cut, and we see that we have three areas: the scala vestibuli, the cochlear duct, and the scala tympani. Let's not worry too much about all these details because this is the basic anatomy course, but the vibration of that is transmitted to the cochlea through the stapes will create an agitation of the liquid inside of the cochlea, and will eventually be transmitted to the cochlear duct. So you see a zoomed in idea of the cochlear duct there, which has this tectorial membrane and a basilar membrane. And in this structure, you'll find the receptors for sound, which are the hair cell. There's something called outer and inner hair cells. We are not going to worry about it for now. Just think about hair cells. So from here, we generate a change of activity in the hair cells. The axons will form the nerve, and the nerve will project, and now you have to connect this with all the pieces. It will project to the pons and the medulla, and then from there to the inferior colliculus, or colliculi because it's two, and then from there to the thalamus, the medial geniculate, and from there to the primary auditory cortex. So there is a story here. I just wanted to make clear that I did not explain very well all the details, but about the functioning, because it's exactly the kind of thing that we need to decide what to teach and what not to teach. So otherwise, this becomes an anatomy physiology course. So there's a lot of material. But it's inevitable that we need to cover some sort of physiology. Otherwise, you would not understand the full details. So when you study, you basically can do the same thing. So when you put all these pieces together, and as a way of also memorizing, you can go over the different pathways. So you start even in this picture, a very good picture. Try to understand how the auricle amplifies and funnels the sound the sound waves onto the tympanic membrane. This vibration is transmitted through these three ossicles in this order: malleus, incus, stapes. Stapes projects onto the oval window of the cochlea, and in the cochlea we have the membranous labyrinth inside of the bony labyrinth, and two sorts of fluid which is spelled out here, the endolymph and the perilymph. So eventually this vibration is going to affect the cochlear duct, which is this middle space when you do a section like this of the cochlea. So you have a scala vestibuli, top one, the tympany below, and then the cochlear duct. The cochlear duct has something called the basilar membrane, where a bunch of cells are resting, so to speak, are lying on top of, and those cells, the ones that we need to pay attention on, the hair cells. The hair cells are the receptors for sound. And there's something on top of the cells that is the tectorial membrane. So it's the movement of these particles that eventually affect the hair cells' activity, and from the hair cells we have the formation of the vestibulocochlear nerve, and then you go through the layers of medulla and pons, thalamus, and eventually the primary auditory cortex, which is in the temporal cortex. And that's the basic picture for an intro to anatomy course. The vestibulocochlear complex also has a vestibular complex. A vestibular complex is an equivalent structure to the cochlea, in which we have cells, there are receptors for the vibration, the movement of the endolymph. And they will be detecting the position of the head in relation to the body. So we have two components: the semicircular canals, which are basically essential for rotation of the head, and the utricle and the saccule, which are important for linear movements. So, for example, nodding yes with your head. And this is as much as in a simplified way that I would like you to know about the vestibular complex. And looking at this picture, it's pretty much what we would stay with. But we'll expand this with a lab textbook, and that offers a little bit more explanations, and you can develop this on your own. Moving finally into vision, and here we are not going to dissect, given that our course now is online, dissect the eye in the lab, but so you have to pay attention to some videos I have there. As much as I know that the sound doesn't come out very well, at least you will have a certain you can always read the captions, let's put it this way. So here it is, the anatomy of the eye from the outside. So there's aspects as to the opening between the eyelids that we can discuss. So that opening is the palpebral fissure. There's a medial angle and the lateral angle. And then you have the sclera, which is the white part of the eyes. And what you see there, you will understand this in a second, you see several layers, but the one that is closest to you is actually the cornea. So the more superficial layer that you have there covering the iris, so the the aspect of the eye that has that color is the cornea. And so everything you see, so the iris, which is the colorful part, it's behind that. But that will be clearer as we go along. So what we're seeing here is exactly that. So this may be a very complicated picture. You say, this is too complicated, I need so much time. Yes, pause here. But again, this may be a good picture for you to come back once we cover everything. We are surely going to simplify everything for you. So first, let's talk about the 3 layers of the eyeball. There is a fibrous layer, a vascular layer, and the inner layer. The fibrous layer is the outermost layer, and we have the sclera and the cornea. The innermost layer is the retina, and the layer in between is called the uvea. So let's start with the fibrous layer. The fibrous layer is the outermost layer. It consists of sclera and cornea. You see the sclera is the white part and the cornea is the most anterior. So that's what we saw from the aperture, the pulpal fissure. The vascular layer has different components, so it's represented in this picture as yellow. So it has the choroid, which is highly vascularized, so a lot of blood vessels, and it continues anteriorly into a structure called the ciliary body and the iris, which gives the eye color. Note that the ciliary body has a muscle called the ciliary muscle. The ciliary muscles can contract and relax, and by doing so, they'll give more power to the lens. The lens is represented that white portion in the middle. So they will relax it or make it more spherical. And that changes the power, how much we can converge. Note that the iris has the 2 muscles that are going to be responsible for constricting the pupil or dilating the pupil. The pupil is the opening of the iris. So the opening that is limited by the iris. So we have a dilator pupillae that when it contracts, dilates the pupil. That's an action of the sympathetic. And you have a sphincter of the pupillae, which when contracts, constricts the pupil. The last layer is the inner layer. The inner layer has 2 layers, a pigmented layer and a neural layer. We are going to see how this all works now, and then we'll discuss a little bit about the neural layer. So see that light enters through the eye, so the pupil is the aperture, so crosses the cornea, will cross, I'm not going to give you the name right away, but I can, it's written there, the anterior cavity, and it crosses the lens to go into the posterior cavity. And that light needs to stimulate the photoreceptors, which are in the neural layer of the retina. So let's see how the retina, the neural layer of the retina is organized. So we have the photoreceptors, which are outermost. which are the rods and cones. The rods are for night vision. The cones are for color vision or daytime vision. We see color when we have light. And then, once stimulated, these cells synapse with bipolar cells and then ganglion cells. The ganglion cells are the ones whose axons will form the optic nerve. So you see that chain: photoreceptors, bipolar cells, and ganglion cells. In addition, there are cells that have multiple synapses: the horizontal cells and the amacrine cells. I would like you to know a little bit more about the photoreceptors. First is understand that they are all over the retina, but there is an area of the retina that's called the macula. The center of the macula is called the fovea, and in this area we only have cones. So this is an area that has very higher density of cones, and it's the area that gives us the best visual acuity. So when you look at an object, choose an object wherever you are, everything else becomes kind of blurry. So what you're seeing very focused is the portion of the image that is falling onto the fovea. Everything else loses detail because it's relatively more and more peripheral. Note that there is one area of the eyeball in which there's we call the blind spot, because there are no photoreceptors. That corresponds to the point where the optic nerve is exiting the eyeball. That is the blind spot. You can choose or find online certain tests to find your blind spot. Very easy to get. It just requires a little bit of concentration. So the eye is divided into cavities. So there is an anterior cavity and there is a posterior cavity. The anterior cavity is in front of the eye, and the posterior cavity is behind the lens. The posterior cavity has something called the vitreous body. It looks like gelatin. It feels like gelatin. The anterior cavity can be divided into two chambers: a more anterior, which is anterior to the iris, and one that is posterior to the iris is called the posterior chamber. The anterior cavity is filled with aqueous humor. So a humor is a liquid that resembles water, whereas the posterior cavity is a vitreous body because it's a gelatin kind of structure that resembles glass. This video will make it is a dissection of a sheep eye, and so this may note that it's not going to be as beautiful because the eyes has been conserved in a certain liquid, so they're not as beautiful as, let's say, fresh eyes. So let's see what it looks like.
Let's cut the eye into a frontal section so we can look at the structures of the posterior segment, which is located behind the lens. Here we can see the back of the iris and the pupil. The The dark area surrounding the iris is known as the ciliary body. The ciliary body helps to anchor the lens in place through the suspensory ligaments. Taking a look into the posterior half of the eye, this object is the lens. You can see that the lens is attached to a gelatinous sac. That's called the vitreous body. The vitreous body fills the posterior segment. It's in contact with this delicate yellow membrane. That's the neural layer of the eye, known as the retina. The retina contains the optic disc, which is where axons of the photoreceptor cells leave the eye, forming the optic nerve, which is cranial nerve 2. The optic disc is known as the blind spot because photoreceptor cells aren't present here, and therefore visual information cannot be received in this area. The retina, which is the neural layer, is in contact with the vascular layer, called the choroid. And the choroid is in contact with the fibrous layer, the sclera.
Okay, there's some kind of components there, a couple of things that are not perfect in the video, but she does have she did add one component that I did not tell you. It's on the slide, but I did not tell you. So let me just go back. So I spoke about the blind spot, but the area that exact spot. So the blind spot is a functional spot, meaning that it's the spot that will no longer see. But anatomically, that area looking at the eye is called the optic disc. So we already covered this component a little bit. So we see if you cover your left eye, you have a notion what you see. You see great part of the right, but you also see great part of the left. So we know that both eyes see something. So how do the image projects onto the eyes? The image projects in a way that is reversed and inverted. That's again is more functional, but it may help. Reversed and inverted. So the right side flips to the left of a picture, and the left flips to the right, and the up flips to the bottom, and the bottom flips to the top. What happens is that each eyeball is capturing simultaneously something of that visual field and something of the opposite visual field. And then, so that our right hemisphere gets only the right portion, the left portion of an image, and the left hemisphere gets the right portion of an image, we have to organize the axons. And that will happen in the optic chiasma. So the first picture, this is naturally more functional, so I'm not extraordinarily worried if you don't get all those details, because you will have physiology and you'll expand on that much more. But you see there is something of the image that the right eye sees it only, the left eye sees it only, there's a part that both eyes see it. And so the light projects all the way to the retina where the photoreceptors are. From there, we need to cross the information that needs to be crossed so that the right hemisphere gets the left part of an image and the left hemisphere gets the right part of an image. That happens at optic chiasma. From there, the fibers will project to the thalamus. We have the lateral geniculate nuclei, sometimes called the lateral geniculate body, and from there projects to the occipital cortex. But signals related to light are also projected to other structures, such as the superior colliculi and, for example, the suprachiasmatic nucleus, which then feeds into the pineal gland and allows the pineal gland to produce melatonin based on the amount of light. A lot of light produces little melatonin, little amount of melatonin. Little light produces more melatonin. And that's pretty much the view of the visual pathways. Now you can put everything together. You can, once you see the problem, not the problem, but the aspect we need to consider is that neuroanatomy needs to start somewhere. So until you know all the names, it's always difficult to understand the full mechanisms. But by now we finished both lectures, so you know all the names. So now you can study this in a more narrative kind of way. Instead of studying the eye and then studying the visual cortex and then studying the thalamus, you can study all together, for example, for the visual pathways. I have 2 more videos to show you about the dissection of the eye. I hope they help.
On the superficial surface of the cow eye, you can see some skin and some musculature still attached. We can also see cranial nerve 2, the optic nerve, on the back. The fibrous layer is the outer layer of the eye. It consists of 2 parts. The sclera, which is the white of the eye, and the cornea, which is the transparent portion that light enters into. It looks gray here because this is a preserved specimen.
Okay, another video and the last one.
The eyeball is divided into 2 segments. The anterior segment in front of the lens and the posterior segment behind the lens. The anterior segment has 2 subdivisions, the anterior chamber and the posterior chamber. Let's remove the cornea of the eye and look behind the cornea into the anterior chamber.
Okay, note that she says segment, but you can also say cavity, like we said. Appreciate what's coming out of the anterior chamber.
You'll notice a clear watery fluid leaking out of the incision. This fluid is the aqueous humor, which fills the anterior segment. The anterior chamber is the space between the cornea and the iris. The iris is the colored part of the eye. It's a muscular structure that controls the size of the pupil. Let's put a piece of white adipose tissue behind the iris so we can better see the shape of the iris. And the opening in the middle, which is called the pupil. Light passes through the pupil to the lens. The pupil leads into the posterior chamber, which is the space behind the iris, between the iris and the lens.
Okay, so these videos will help to a certain extent because they allow us to see the cow's eye, but note that the eyes are not fresh. They've been conserved in a certain liquid, which alters a little bit about of the anatomy.